field and looking at diploid canescens and tetraploid austrinum. The thing to know is that in the 70s, Clarence Tao had found triploids in tetraploid calendulaceum, but it wasn't until 2006 that Dr. Ranney discovered that austrinum and atlanticum and colemanii, as well as calendulaceum or tetraploids. Up to that point, these three species were considered to be diploid, but starting in 2006, we knew that wasn't the case. At the same time, Ron Miller in 2006 found out that austrinum could be pink as well as yellow. So two things happened in 2006. Austrinum became a tetraploid, although it obviously had been all along, and it could be pink as well as yellow. The way Ron went about determining the difference between austrinum and canescens in his area around Pensacola, Florida, was that austrinum has glands on the new growth and canescens does not have these glands. So what does canescens look like? Well, it's a diploid and it looks pink. Almost anyone who's looked at deciduous azaleas knows canescens, especially if they're in the South. It's a very common deciduous azalea. Austrinum, this is the typical austrinum. It's this orange-yellow. And I said, up until 2006, everybody thought austrinum basically looked like this. But Ron discovered that austrinum could also look, it could be a pink color with yellow blotches. But it can also be just pink. So the question came down, how do you tell an austrinum from a canescens? Well, Ron answered that question. The austrinum will have glands on the new growth and the canescens will not. So what do we mean by glands on the new growth? Well, as you can see here, here are your glands. There's glands on the leaf, there's glands on the leaf bract, there's glands on the stems. If you saw a canescens under this kind of microscope, you would not see those glands. Mossman in the 1970s talked about the fact that occidentale was a poor pollen parent. And this is what Sally and I's initial question was. Why is occidentale a poor pollen parent? Well, what we discovered in 2010 is it has nothing to do with occidentale as a species. It has to do with occidentale being a diploid. Diploid cross tetraploid produces seed pods, but tetraploid cross diploid does not. This was a key part of our findings that allowed us to go in the field and look for triploids. The other thing we discovered was that when you cross diploid by tetraploid and you get seedling, the physical characteristics are dominated by the tetraploid pollen parent, not the seed parent. So these triploids are going to be almost identical in appearance to the tetraploid pollen parent. The other thing that's been known for a long time is when you cross diploid, cross tetraploid, you get triploids, and these triploids are normally seed sterile. In the 2020s, Samantha Pryor discovered, as well as other people, that for deciduous azaleas, your butterflies and moths can fly 5 to 20 miles to pollinate. So your pollinator can actually be quite far away from the plant that's being pollinated. So what are we using as our idea of finding suspected triploids? Well, they're not going to be near the austrinum. They're going to be near the canescens. So your triploids are going to be near your canescens because it's the canescens that's going to be the seed parent because it's the diploid, and the tetraploid austrinum is going to be the pollen parent. You're not going to get triploids in the other direction because we showed that by hand pollination. The other thing that we use as an indicator is we found our first triploids, suspected triploids, by looking at places where there's a large amount of canescence and only a small number of plants that look like austrinum. So if you have a large number of canescence and you walk up and you find austrinum types, then those austrinum types are very likely to be triploid. So based on this, we've found 14 locations for austrinum-like triploids. They're in the first one that was found was in Albany, Georgia. And then we worked with Lily Bird to find some near Tallahassee. There was an old finding of a unusual plant at Mariana in a population of austrinum and alabamiense. It turned out to be triploid. And then over in the Pensacola area, we found a population. So the first 4 populations found went from Albany, Georgia, over to Pensacola. So that was year 1 of finding those, and it was all happenstance that we found the 4 locations at one time. The next year, we found 6 locations in the Blackwater State Forest, based on Ron Miller's information about known populations of canescens with yellows in them. And then this year, we found 2 more locations in the Pensacola area in the Blackwater Forest, but we also found some in Geneva State Forest in Alabama, and up near Midway. We by looking at iNaturalist, we contacted someone who had found an austrinum type up there, and when we looked at it, it was off river, and we decided it was worth testing, and we tested it and found out that it also was a triploid. So what do triploids look like? Well, as we've said, these triploids are going to look like typical austrinum. And in fact, it's a funny statement would be they look more typical austrinum than austrinum actually does, because they tend to be these classic yellow flowers. But they don't have to be, just like austrinum, they can be these pinks with blotches, or they can be pure pink. In other words, they're following their tetraploid pollen parent from the yellow to the pink with blotches to the pinks. And in fact, looking at this picture, I do not believe you can stand 10 feet away and tell a diploid canescence from a triploid from a pink austrinum. And in fact, we know locations where within a few hundred feet of one another, you can find a pink canescence, you can find your triploid, and you can find your pink austrinum. The key here is you can't do it based on appearance. What we're really looking at is we knew 2 things to be true. We knew the triploids were going to be close to the canescence, to the diploid, and we knew they were going to look like the tetraploid. So when you have a yellow austrinum and a pink canescence, it's pretty easy to see the triploids because it'll be yellow. But when you have a pink austrinum, then it becomes harder. Now, how do we tell the canescence from the austrinum? The glands on the new growth. The austrinum will have glands, the canescence will not. The triploid will also have glands on the new growth. So here's a triploid that we found at Goose Pond. Now, Ron's known about this for a while, but Goose Pond has 1 yellow and hundreds of canescens. Classic spot for a suspected triploid based on our rules. Lots of canescence, 1 yellow, but here's what Ron said. As I like to say, every week we know less and less about azaleas. By next year, we may know nothing at all. Just look at the attachment, an absolutely beautiful austrinum from the roadside near Goose Pond. A perfect example of an austrinum at its best, except it is a triploid. a singular yellow triploid in a population of pink diploid canescens created by austrinum pollen from afar. So this population actually contains no tetraploids, but on the river nearby, there are tetraploids. And we're talking here probably 5 miles between the river and this particular location. We know of a place at Wolf Creek, which we knew about in 2010, which also has a huge number of canescens and what we thought in 2010 was an austrinum, but we found out in 2022 was actually a triploid, but using flow cytometry. And that thing is 13 miles from the nearest known tetraploid location. In summary, triploids are common, yet it took us 15 years to find an austrinum-like triploid. Triploids are created whenever there is overlapping bloom time. Triploids are created even when no tetraploids are in the immediate area. Triploids are normally near the diploids. That's who produces the seed for the interaction. The triploids are hard, if not impossible, to tell from the tetraploids. Triploids will normally be seed sterile. Triploids can be fertile. Diploid cross tetraploid can produce fertile tetraploids. We know that from hand crosses. We also know that triploids can be fertile from hand crosses. Such interaction almost certainly occurs for all of the tetraploid deciduous azalea species. But it also almost always occurs for any rhododendron population where there's interploidy contact zones. This is incredibly important for people who are studying lepidotes or virea, both of which where we know by hand crossing, lepidote triploids are very fertile in general, and virea, based on cytometry scores, we know that the polyploid interaction can occur there. So this result should in fact be able to be done in the field for lepidotes and virea. What we have here is the thank yous that we have to do, because this has all been confirmed by flow cytometry. Mariana Castro at the University of Cambrai and Arzaro at the Florida International University have made this study possible. What you're seeing here on the right are actually the latest results. What you'll notice is the scores around 2.4, 2.5, those are triploids. The scores in the 3.2 area are tetraploids, and the scores in the 1.6 area are diploid. And we predicted this. If you'll notice this chart, everything that says CA turned out to be a triploid. The things that say C, that's for canescence. Those turned out to be diploids. The things that are called A for austrinum turned out to be tetraploids. And we collected a colemanii, and it turned out to be a tetraploid too, as we would suspect. So we showed in this latest example that we could send in 14 examples and know the triploids from the tetraploids, but we did this based on the rule of going and looking at a diploid, looking at the canescens, which is a diploid, looking for austrinum types next to them, looking to see if they were seedless, and then that made it a suspected triploid. And this seems to be working from Albany, Georgia, clear across to Pensacola. It probably wouldn't work in Mississippi, though, because in Mississippi, austrinum doesn't have glands. But from Pensacola to Albany, Georgia, for all we know, austrinum typically does. So if you want to see pictures of this phenomena, on iNaturalist, we have an observation of a boat ride on the Escambia where we look at pinks and yellow tetraploids, and we have a walk around Dantley Branch where we look at austrinum, where there are pink diploids and triploid austrinum types. Thank you.
Thanks, John. You have a question from Michael Bamford in the Q&A.
Okay, in the Q&A.
So if you click on it, I'll read it so everybody can hear it. Are you delineating the species based on the key code, glands? Do you think the legacy key codes will hold to be an accurate delineation in the future?
Okay, so we base most of our observations on the classic key codes, but what we're really looking at is what we know about diploid and tetraploids. We spend most of our time thinking about things in terms of diploid and tetraploid, not in terms of whether they're canescence, but whether the canescence is a diploid, and austrinum not as of austrinum, but as a tetraploid. Then we use the local characteristics, as I said, in the Pensacola area. Austrinum does have glands, and canescence doesn't. Now, we just know that from a lot of flow cytometry that's been going on since 2006 by various people, including us. So do I think the classic codes are going to hold up? I think we're going to have to wait for the DNA analysis that's being done in Germany to know whether how well our eyes are telling us the species and how well the keys have done it. But we do know that in the area that we showed on the map there, that for canescence and austrinum, the key thing is looking at glands on the new growth. That's the key. Color doesn't help. I can show you miles of pink austrinum, and the normal person, if they use the key, a normal key, would key it out as canescence, because if you look at Crone says canescence can have glands on the new growth, which it actually can, because there's canescence in Texas that have glands. But in general, in the Pensacola area to Albany, Georgia, that is not the case. Hope that answers the question.
We have a question from Dylan Nakaji Conley. Dylan, go ahead with your question.
Hi, sorry if you've already explained this, but what do you plan to do with this information on the potential misidentifications? Do you intend to relay this information in a public forum that informs botanists of this possible misidentification that you found?
That's an extremely interesting question. What we're trying to do as uncredentialed field people is tell as many people, like in the Rhododendron Society and in academia, that what they classically think, in other words, an iNaturalist, if you go to iNaturalist, every pink in the South is a canescence, which we know for a fact, if you're on the Yellow River or the Escambia River or the Blackwater River, that's just not the case. Those pinks on the river are, in fact, austrinum, pink austrinums. How we get that message that we've known since 2006 into the academic world and into the general rhododendron world, and into the nursery trade and so forth, I don't know what the forum is. I'm hoping that Juliana can be a mechanism there. As far as the triploids are concerned, that's all happened since 2022. There's probably like 6 people in the whole world that even care. And there's probably like 4 or 5 people that can actually go out in the field and make an educated guess. I think the first thing is the pink austrinum being accepted. And Ron did write an article on the azalean about that. But again, how you get that into academia, I don't know. So my short answer is, we're trying to get as many people, anybody who wants to come to Florida and see pink austrinum and triploids. If you come down to the Pensacola area in February to the middle of April, Sally and I will be more than happy to take you out and have you physically touched pink austrinum and suspected triploids? Does that approach your question?
Yes, thank you.
Thanks, John. You know, this is a mechanism that I'm hoping will help get more attention on the interesting mysteries that I think your work really speaks to the interesting in-depth work that needs to be done in this system. And I could see a lot of work following from these patterns that you've shown. So we'll keep talking about it. I have one question really quick before we transition, and I don't want you to fully answer this because I think it's a big mystery. But we have a mystery plant here at Holden that it shows that it's a wild collected plant, and it shows that it's a calendulaceum. But when it flowers, it has orange, yellow, and pink flowers. It doesn't look like a calendulaceum.